A lot of my research involves studying protein-protein and protein-nucleic acid interactions. And so today I wanted to just go over some of the techniques that we use to address these type of questions and how you might go about approaching studying a protein-protein or protein-nucleic acid interaction. So thinking through some examples and that sort of thing. So it's going to be a more technical post, less background than usual. I'm going to, in the blog version, you'll be able to find links to the various techniques that I talk about if you want to learn more. Today is more just kind of like an overview and just kind of getting you in the mindset of thinking through how you might approach some of these problems. So here is a basic outline of where we're going. We're going to talk about some different techniques, as well as think through an example and talk about how we could go about actually getting the molecules we want to study. So our premise is that we have some molecules and we want to see how they interact because molecules are really cool on their own, but it's really when they come together that the real magic of biochemistry happens. And so there's a bunch of different techniques that you can use, and it all kind of depends on what answers you're trying to get and what your like study system is. So are you studying what's going on like actually inside of cells or inside of animals or whatever? I'm not really going to talk about the animals, but like inside of cells or that sort of thing. As you could do some of the stuff with like tissues instead of cells, but I don't like dealing with actual like living things other than like bacteria and insect cells and that sort of thing. But anyway, so that's going to be like your system or whatever. So that would be more like in vivo or in cellulo or in cells or whatever, just like, or if you're doing it in vitro. So typically when we talk about in vitro, we're talking about like using like purified components, or at least like semi-purified components or that sort of thing, where you're mixing these different components and you have more control over the system. And so typically most of the work I do other than like expressing my proteins and stuff is in vitro. And this is most of what I'm going to talk about, but also tell you about how you can study these things, some of the techniques that you use to study these things in like inside of cells and that sort of thing. A lot of times people will do like a combination of both. So it's important that to recognize that there's benefits of studying things in cells and there's benefit when people are cells and like organisms and stuff, but there's also benefits in studying them in vitro where you have more control and you can get at more like granular mechanistic details. But if you want to, sometimes if you're looking like too narrow, you're missing the bigger picture. And sometimes there could be extra components that are missing in vitro. And so things might not behave quite the same in vivo. And so you can kind of like address these questions in your in vitro system and then test their importance, see if your hypotheses make sense in the larger context in the actual like cellular environment. So we have a protein and we want to see if it's interacting with other proteins or if it's interacting with other nucleic acids. And so we can start from the point where we don't know what's interacting with what, or we can start from the point where we know what's interacting with what, and we want to study it further. Even if we know what's interacting, we might not know where they're interacting. And so the real key thing is that ideally you'll figure out that two things are interacting and you'll know like where they're interacting. So like where on each molecule is involved in the binding. Then you can mutate that place. So either on both things or one thing. So ideally, you know, like this place on the protein is binding to this place on the nucleic acid or that sort of thing. And then you can mutate them. So you can change those amino acids in the protein that you think are responsible for binding, or you can change the nucleic acid sequence where you think it binds. If you change either of those, you should lose the affinity for the binding. You should, and then you're like, yay, I was correct, they were interacting there. You don't always have this much information. There are ways that you can get to more information about where they're binding, and so we'll talk about some of those things, but this is kind of like the ideal situation. And so, of course, to measure this, you're going to need some techniques to talk about how you can measure these interactions. And so we'll talk about those sort of things. And then we'll go through like an example of really thinking this sort of thing through. But let's talk a little bit more about the methods. So we have these different ways to study things, like I was saying, like some of them are like in cells and in vitro. And so we'll start with the in cell stuff. So often, unless you're like testing your hypotheses, like after you do stuff, a lot of times when you're working with like in cells, you do a lot of that work is often more kind of exploratory. So maybe you have an idea and you want to test it. You have an idea it might interact with this sort of thing, but a lot of times too, you don't really know what it's interacting with, and so you want to find out. So there are different ways to do this sort of thing. So a common way if you want to study like protein-protein interactions is like mass spectrometry. So basically this is like you take your cells or whatever, and you often do this with like a cross-linker, or you do it with biotinylation. So like this BioID technique. So basically you can have like one protein or something of interest, you can attach it, like conjugate it to a biotinylating enzyme. And so this is going to put a biotin tag on anything that's nearby. And then you can then like isolate with like streptacoccus beads or something, isolate the biotinylated products and then sequence those, mass spec those in order to get information about what that was at least near. You can also cross-link things. So typically with the cross-linking, if the two molecules are close enough together, hopefully directly interacting, then they can get cross-linked together. You can isolate those products and identify them. And so these would be kind of mass spec based result. And so you can basically you can isolate, you can pull out the protein of interest so that you want to see what it's bound to. So in the case of like the BioID or whatever, you would have that protein of interest, you can conjugate it to the biotinylating enzyme, and so that would tell you that the things that are biotinylated were like bound to it or nearby. You can do something similar if you're doing like a mass spec, you can pull out that protein of interest. So you can either with like an antibody against it or some sort of thing, if it's like tagged, you can pull out the tag. And then since you're isolating that protein specifically, then what you're sequencing is going to be that protein and anything that's bound to it, although this binding might not be direct. So these are important things to keep in mind, and why it's important to then further characterize the interaction in vitro and see if they really are interacting, because you can imagine that if you had like a multi-protein complex, you pull out your protein of interest, but it's bound to something that's bound to something else. And you do the mass spec, unless you wash this super duper thoroughly, in which case you might wash off the actual direct binding partner, then what's going to happen is you're not going to be able to tell if these were directly linked to one another, or if they were just nearby, if they were just pulled out alongside of what was directly bound. And so if you're wanting to test if something is directly bound, then this is where we're going to talk about doing like mutations in with the part that we think they bind. And if that is really where they're binding, you're going to lose the interaction. Or if you purify the components separately and mix them together, do they actually interact? And so these are things we're going to talk about when we talk about the in vitro techniques. So for example, if you have these molecules, they're interacting, you can cross-link them together. They have these special mass spec cleavable cross-linkers, where basically you can isolate the cross-linked peptides and then determine actually where they're cross-linked with mass spec. And so this can even give you more information than you get from just knowing that they were bound together and that sort of thing. You can do something similar when it comes to nucleic acids. So say if you're wanting to look at if it's binding to like DNA or RNA, you can do you can like cross-link it to the nucleic acids. Some methods use like UV for like linking to RNA. Methods use like formaldehyde and other linkers for linking it to DNA. So a common version of this is ChIP-seq. And this is often used to study regions of DNA that are like having different levels of transcription and that sort of thing, or different levels of various factors bound. So basically in that case, instead of using mass spec to identify the proteins that it's bound to, what you're doing is you're using sequencing. So you're sequencing the nucleic acid to see where the specific protein is bound. And so you can immunoprecipitate, so use like antibodies, or you can do some other sort of pull down where you have some way to isolate a protein of interest. So either an antibody against that protein, or you have some sort of thing that's bound, the protein is tagged, and you can pull down the protein specifically, and then sequence what it's bound to. And so this is going to tell you about like where a specific protein is bound. If we have an idea, so it's like the mass spec or whatever, we're kind of just going into it like, what is this bound to? With so basically it chops up the different proteins into peptides, and it searches a big database to try to analyze the different peptides and see what sequences they correspond to, what proteins they correspond to. If you have an idea of what it's interacting with, then you can do a Western blot. And so basically with a Western blot, what you're going to do is you're going to run an SDS page, separate the proteins by size, transfer them to a membrane. And then what you're going to do is you're going to use antibodies to probe for a specific protein. So if you do something like a pull down where you take the protein and you pull down anything that it's bound to, so you isolate the protein, and when you pull it out of the cells or whatever, it's going to be bound to other stuff likely. And so with the pull down or whatever, you do some washes, but you don't do it like super stringent, or else you'll lose everything. But you want to have at least like it's important that you have like a high enough salt concentration that like the really weak interactions are going to get lost. So you're not getting super duper like non-specific or really, really indirect interactions, depending on what you're going for, of course. But the basic idea is that the things that are bound to that protein are going to come with it. And then if you have an idea of what's there, what you think it's bound to, then you can test if it's actually bound to that thing using antibodies against the protein that you think it binds to. And so the difference here is that we actually have to have an idea of what it's bound to. Another way that you can study protein-protein interactions in cells is using something like a yeast-2 hybrid or similar. These methods basically rely on some interaction occurring between the two proteins that typically often like attached to 1/2 of something. And then when these components come together, when they bind together, then you get the active component. So this can be like activate a gene expression, or it can make like form full GFP. So you can have like GFP fusion type things where you have like half of the GFP here, half of the GFP here. If these proteins come together, the GFPs come together, and then you get glowing. So there's different things that you can do to study interactions in sort of cells and that sort of thing. In that case, you also need to know what's going to interact, although you can do like screens where you basically have one protein, and then you attach the protein, the like second half, to a bunch of different proteins and then test which of them. So there's more like high throughput screening type things that you can do as well. So those are the basic things about doing it in cells, but I want to talk more about doing things with purified components. This is most of what I do. And with anything, I forgot to mention that there's a lot of variations on all these different techniques. There's no one right way to do anything. And so a lot of times it's going to depend on like what your lab has in terms of equipment, in terms of funding, what kind of questions you're asking, what techniques you are most comfortable with, what techniques people in your lab do, all sorts of various things. And so there's no one right answer. Some techniques are better for different things than others. And yeah, so this is not a comprehensive list. These are just some of the basic strategies that you'll hear talked about and that you might encounter if you are doing this sort of research. We're going to talk about some different methods for studying these. So for protein-protein, we'll talk about like analytical size exclusion chromatography, native page, some biophysical techniques. For nucleic acid proteins, we'll talk about like slot blot filter binding assays, EMSA, some other biophysical techniques, footprinting. We'll talk more about all these different things, but in order to do these types of experiments, we need to have purified components. And so how are we going to get these components? So for nucleic acids, this is easier typically. So for short ones, we're talking we normally order them synthesized. And so you might have had like primers synthesized, so like short pieces of DNA. The DNA is usually much, much cheaper than the RNA because RNA has that 2' OH that makes it less stable and harder to synthesize because that group has to be protected and all this different stuff. So your RNA is going to be way more expensive than your DNA. But you can still order it like short bits of RNA custom made, like synthesized with like solid state synthesis. It uses things like phosphoramidites and stuff. It's pretty cool stuff. But anyway, that is if you want to do like short pieces. For longer pieces, so for DNA, what we can do is basically we can clone the sequence we want into like have copied into like a plasmid, and then use either PCR or stick it in bacteria to have them make it for us, make lots of copies of it. Then we can purify out those copies. You're probably familiar, likely with doing like a mini prep to isolate plasmid DNA. So basically we can do these various things. Then if we like design it so we have like restriction cut sites, we can then cut out the region of DNA we want. If we're doing it from plasmid or if we're doing PCR, then we just have these linear fragments we can then isolate with like a PCR purification. For RNA, we use like in vitro transcription. So in this case, we're going to start with it with the DNA, and then we're going to use a DNA-dependent RNA polymerase. Usually we use T7 RNA polymerase, so it's from this like T7 phage. We put the T7 promoter in front of it, and we get lots of RNA of this sequence made. And so we can do it like in vitro, so outside of the actual bacteria, and then we just purify it and use it. And so this in vitro transcription is a super duper useful strategy. And this is like what is used to make like mRNA vaccines. When we're working with nucleic acids, often we want a way to track them. So what we often do is we radio label them at the 5' end typically. It's a lot easier to label it at the 5' ends. So the 5' end normally has a phosphorus group. We can replace that phosphorus with a radioactive phosphorus. So typically P32. And then basically we're going to have this it'll radioactively decay. And so this will give off signal. A lot of people nowadays use like fluorescent labels for some things. Fluorescent labels can be great because you don't have to worry about the radioactivity, but they're less sensitive and they're also bulky. And so we still very often use radioactive labeling. So you can label at the 5' end, you can also body label, so label like the entire thing. Various things. And so the great thing about this radio labeling is it's super duper sensitive. You can use really, really tiny amounts and be able to detect them. And you can, it's non-invasive type of thing. Like it's not going to alter the properties. It's not going to make it bulky and that sort of thing. So you're better at mimicking what's actually going on without artifacts.
when it comes to the proteins, this is going to be more complicated. So we're going to typically do some sort of recombinant protein expression. So when we're talking about recombinant, basically we're recombining the genetic instructions for the protein we want with another piece of DNA, this like vector, the backbone, that's going to serve as a vector or a vehicle for getting that into cells to express it. And so often we put it in like a plasmid vector and we can express it in E. coli. E. coli works great for some things, but not great for other things. And so you might have to experiment. So bacteria typically, they don't do as well with like bigger proteins or proteins that need help folding or need get post-translationally modified and that sort of thing, because bacteria don't have the same, they don't do that sort of thing. They don't have the same helper molecules that we do and the same post-translational modifiers that we do. So if you want to study something kind of like more complex or whatever, you want something more, typically we're studying human proteins, not always, and so if we want something more human-like, we go higher up in the eukaryotic hierarchy or whatever. And so we can use yeast cells. I've actually never used yeast cells. Apparently they're really hard to break open to lyse. A lot of people use yeast for various things. I typically use insect cells, and so we use like a baculoviral expression system. I post on that if you want to look. And then also like mammalian cells, so like hex cells and that sort of thing. And so as this slide suggests, and I post on this problem suggests, there can be, you can have difficulty with protein expression. So there's typically going to be a lot of optimization in both the expression and the purification steps. And so we just talked about there can be expression problems, but there can also be purification problems. And because we need to get these proteins pure, because we're expressing them in cells, and it's a lot harder to purify a protein than it is to purify nucleic acids, because we need to be a lot more like picky kind of when we're getting these proteins pure, and different proteins have different ways that like different properties, and so we have to really optimize to get the protein that we want and not the proteins that we don't want. And so typically what we're going to do is we're going to one of the ways that we can help is by adding a tag. So at the N or C terminal, typically the N terminal. So this is going to be like an affinity tag that's going to help us with purification, and it's also going to allow us to like pull down the protein like we were talking about earlier. And so some common tags include like the strep tag, the his tag, GST or HA. Sometimes we have like solubility problems, so we add a fusion partner. And so this is like a little protein that's going to get made, and kind of the cells like making it, so it'll make it first. This is going to kind of help drive the making of the rest of the protein. That's going to help keep the protein soluble. And so typically what's going to happen is you'll put this tag, and then you'll have like a cleavage site for a protease, site-specific protease like TEV or precision protease. So you can then cut this tag off when you're done with it. But typically you want to keep that tag on for your first step of protein chromatography. And so the way we purify proteins is usually with protein chromatography. So basically we have these columns filled with resin, these little beads called resin. And this can be in this like gravity flow format or in like an FPLC, like an ACTA machine. And basically these resins can have different properties. These will affect how they interact with the proteins. So if you flow a mixture of proteins through these columns, the different proteins will interact differently with the resin, and so they'll flow through at different rates. And therefore, by isolating the fractions, you can then isolate your protein of interest and apart from the other proteins by exploiting differences in the various proteins. And so typically we start with affinity chromatography. We use that tag to take advantage of it. The tag the resin has something that binds to the tag specifically. So you let your protein bind, everything also flow through. Then you can pee it off with something that mimics the tag, but isn't on like the protein. So with like strep tag, we use like a streptactin resin, and then we have this biotin. With the his tag, we use like nickel NTA, where you can pee it off with a imidazole. So there's various, you have there's different like flavors of this depending on your tag. Once you do that, then you can get rid of the tag a lot of the times. If you want to study the protein, it's like most natural form. Then since you don't have the tag, now you're going to separate based on properties of the protein itself, like untagged naturally. So we can take advantage of its charge with ion exchange chromatography, where you have like charged resin, and then you can compete off with higher amounts of salt. You have, and then like size exclusion chromatography, which is going to separate things based on size. So basically these beads have these little pores, and the smaller things can travel through all of the pores, so they have to travel further. And the bigger things kind of can't fit through the smaller pores, so they don't have to go through them. And therefore they'll travel faster, and you'll end up separating the proteins based on their size. So in these ways, you can purify the proteins. You can check for how pure they are along the way using SDS page. So basically you unfold the DNA to the proteins, you coat them with this negatively charged detergent, SDS. Then these denatured proteins are going to run through the gel. The bigger ones are going to get tangled up more, and so they're going to run slower. And so you're going to get bands where each of the proteins are, and you want to see the number of bands decrease as your protein gets more and more pure. And ideally, if you have a really pure protein, you're only going to have like a single band. As we'll talk about later, we often, when we're doing the cloning, we can use methods like slick or quick change for site-directed mutagenesis in order to introduce amino acid substitutions. So make mutations that are going to change the sequence of the protein and allow us to test whether these are actually like certain residues are actually important for the interactions. So let's talk about some of the methods that we can use to actually study these interactions. So for nucleic acid interactions, the method that I've used the most is called slot blotting or filter binding assay, where basically you have this vacuum filter apparatus that's going to help you pull liquid through, and it's going to be pulling it through. So there's a nitrocellulose membrane on top that's going to bind to protein, and then a nylon membrane that'll bind to RNA beneath it. You take your protein or DNA. I do it with RNA typically, but you can also do it with DNA. This will be like radio labeled, so you can track it. What you're trying to do is basically you're mixing the protein and the nucleic acid. You mix different concentrations of one of the components, typically the protein, because you have this radio labeled nucleic acid. You can use a really, really low quantity, and then you can mix them together and see how much is bound at these different concentrations by separating so that the protein and the RNA that's bound to it is going to get trapped on the nitrocellulose. The free nucleic acid is going to flow through, and then you see where the radio labeled nucleic acid ends up. Because you're doing this with a range of different concentrations, you end up getting, you can get information about like equilibrium thermodynamics. And so you get information about the KD, about the affinity. And so if you have something that's a really high affinity interaction, you won't need much of the protein in order to get it all bound. But if you have something that's a weak interaction, you'll need a lot of the protein. And so the point at which half of it is bound, the RNA is bound, is going to be your KD. So much more on this in other posts. You can get another method for studying protein nucleic acid interactions is called an EMSA, or an electrophoretic mobility shift assay. And so basically in this case, you take your protein and your nucleic acid and mix them at different concentrations, just like we did before. In this case, however, instead of separating them like on these membranes, what you're going to do is you're going to separate them on you're going to separate them in a gel. And so when there's no protein the free probe. So your probe, so the probe would be like the nucleic acid you think it binds. It'll be unbound, and so it's going to run really fast, and so it'll be low down. As you increase the protein concentration, if they bind, it's going to shift. The more protein you bind, the more the band is going to shift, because it's basically now, because it's bound to the protein and not just free, it's going to be bigger, and so it's going to move slower, and then eventually it'll all get shifted. And so if you do a concentration range, then you can find like the approximate KD and that sort of thing as well. There are also, so this is telling you about the thermodynamic properties. It's telling you about like the KD, about the affinity. And it's doing it at equilibrium. There are also techniques like SPR or MST. So SPR, surface plasmon resonance. So basically you have these like, there's these chips, they have this like special surface, and you bind one of the things to the chip, so the protein or the nucleic acid to the chip, and then you flow the other component over it. If they interact, then it's going to kind of change the properties of the like surface of the chip. And so when it sends like light, the light, when it interacts, it's going to, because the surface is now changed, the kind of like the angles at which the thing light is going to get reflected is going to change a little, and this can get measured by the machine. The important thing to note is that it's going to give you information about the binding. You can get information about like the binding and the unbinding, so you can get actual kinetic information about like K on and K off. Another one of these like biophysical techniques, MST or microscale thermophoresis. And so basically you have these little like glass tubes, these little capillaries, and then it's going to use like this one of them is like fluorescently labeled, and this technique is going to basically measure kind of these properties of how the properties of this fluorescence when you have like different temperature, and it affects how it moves and stuff. It's kind of complicated. I don't know all the details, but the idea is that if these are bound, it's going to change the fluorescent properties. And so this is going to then influence the signal, and so you can get information about the binding that way. So these are just different techniques. So each has different pros and cons, and each will involve their own sorts of troubleshooting. Importantly, these things are not testing to show you like where something is bound. And so if you want to know like where specifically it's bound, one way that you can do this is with like DNase one-spot printing. So in this case, you would have like your radio labeled DNA. You get your proteins to bind to it, then you chew up with DNase 1, you basically cut the DNA, and then you look to see, you separate all the different pieces, and you look to see what region is protected, so you don't have any like pieces corresponding to a DNA of that length, and that tells you that it's protected. There's also like toe printing techniques where you like sequence up until the protected region. So there's various techniques that you can look at to get information on the actual location of binding. You can also test this by mutating the sequence. And so what's important to note is that when you have, one thing that you can do, so say you have EMSA, what you can do is that, so you're going to get a shift if your protein is binding the nucleic acid anywhere in the nucleic acid. However, what you can do is if you mutate that sequence where you think it should bind, and then you lose the binding, you won't get the binding, and that's going to tell you, yes, you actually were binding in that spot. But you have to know where to make the change, or you have to do some sort of like testing of like random ones, basically somehow narrow down what it is. Another point while I'm on this is that with the EMSA, you can detect like stoichiometry. So how many like proteins is bound to a nucleic acid. But you can't tell if these are bound directly to like two sites or two bound to each other. But if you mutate on one site and you still have some binding elsewhere, that can tell you you have like a second binding site. If you mutate and nothing changes, that could tell you that they're two bound together, or it could tell you that you mutated the wrong spot. And so we'll talk later about how you can make mutations in the protein to test protein binding sites, like this where the protein is actually binding it. But this will tell you about the nucleic acid. And so let's review, kind of just like go through an example of, say you had a protein nucleic protein DNA interaction you wanted to study. So if you mutate that sequence, you should be able to lose the binding. And so we just talked about different ways we can measure the binding. And so in addition to being able to mutate the sequence and seeing that it no longer binds, if you want to see that this binding really is specific and it doesn't just bind any old nucleic acid, there are different things you can do. And so one of the things that's important when we're talking about protein nucleic acid or protein interactions is that you need to make sure you have enough salt. So salt can kind of act as kind of like sort of non-specific competitor. And so if you have no salt, or if you have really, really low salt, basically the protein, the interactions that it'll form more interactions than it normally would. The proteins will kind of, it's because they don't have the salts to help like fulfill their needs, they'll kind of glob onto other proteins in order to satisfy those needs. And so you'll get more non-specific binding than if those other proteins have to kind of outcompete the salt. So you want to make sure you have a high enough salt concentration no matter what you're doing. In addition to that, like super generic kind of like competitor, you can often want to use like some sort of generic competitor. And so you can use this competitor DNA, and if the competitor DNA is binding, then your actual probe sequence that you think it's actually binding to isn't going to bind. It's going to bind a lot weaker. And so if you're able to easily compete off your thing with just like generic DNA, your protein's probably just binding DNA generically instead of that particular sequence. Additionally, so if you think it binds like a super duper specific sequence, and then you mutate that sequence, then you can get like a specific non-specific inhibitor kind of thing as competitor instead of just like a generic competitor. So you can also mutate the protein and see if look at things from the protein side. And so now let's talk about how we can kind of mutate the protein, and then we'll go back to this and some protein-protein interaction strategies. So the key here is that because we're going to be doing like recombinant protein expression, because we're sticking the genetic instructions for that protein, getting that specific protein made, we can make changes in that sequence that are going to make changes in the mRNA that gets made, and then in the corresponding proteins. And so basically we can change the codons. So these like 3-letter words that spell the different proteins. If we change these codons, we can get them to make different proteins. So the methods to do this include like slick and quick change, different methods that you can use in order to like use PCR to make mutated versions that you can then like stitch together. So there's different methods, but the basic idea is that we're making site-specific mutations. And so this is like site-directed mutagenesis. And so the basic idea is, so if you think of you're mutating a specific region to test whether that region is actually important for the thing that you think it's important for. And how you know where to, this is often aided by structural biology. And so structural biology looks at like the interplay between form and function, and often uses techniques like crystallography, cryo-EM, or NMR in order to figure out the molecular structures, the 3D structures of these molecules. And you can find these structures, structural models in the PDB, which I talk about in other posts. The really great things about having these structures is that they can actually show you where interactions are happening. And then you have a place where you'll be able to say, okay, I think if I mutate this residue, if I mutate this, and so by residue, that's just when you join amino acids together. Now they are no longer amino acids because they use their amino and acid parts to join together. And so now you call the remainder a residue, although we often use the terms like interchangeably. Technically, they're residues once they're incorporated into like a peptide chain. But when you have a structure, you'll be able to see regions where you think that they should interact. Then you can make hypotheses. Like if I change that, it shouldn't bind, or if I change that, it shouldn't do whatever enzymatic function I think it does. And so structural biology can really help us develop ideas about where things might be happening in order to test them. Sometimes we don't know, we have like no clue. And so what we can do is we can make truncations. So basically we can cut off parts so that we can keep shortening the protein or cutting out parts to see if it still does what we think it does. And so we can talk about different words, different types of mutations. So we talk about like amino acid substitutions where we're changing letters. We can talk about like, we often talk about truncation. So like we were talking about before, where we're basically cutting it from the end. So to indicate a stretch of removed amino acids, we use this like delta sign. So if we removed 80 to 100, we'd write it like delta 80 to 100. If we make a substitution, we typically write it in this format where you have what it was in front and then what it is now after. So like from glycine to an alanine, that the 91st amino acid would be G91A. We typically call like the non-mutated version wild type. And then we often call the different forms constructs. This is just a little like terminology that you might encounter. An important thing when we talk about any of this is that you need to make sure that the changes you make aren't just messing the protein up. So you want to make sure that your protein still folds and functions normally in other aspects. So if you do something to a protein and then you see it's not binding your DNA anymore, you're like, aha, I found the place where it binds. Not necessarily. You might have just made your protein that's like totally misfold. So sometimes if your protein just totally misfolds, it won't even express. Like the cells will be like, nah, won't make it. Or it'll get expressed but quickly degraded, like partially expressed but then like degraded, or it'll get aggregate together into these clumps, into these like exclusion bodies. So basic things. But a lot of times it might express fine, but then when you go to purify it, maybe it's like a weird looking peak or whatever. That could be a sign that there's something wrong with the folding, that sort of thing. And so basically when you're doing chromatography, like size exclusion chromatography, a good sign that your protein's still folded nicely is if you have a nice sharp peak. But some proteins kind of have wonky peaks all the time. Can be due to modifications, various things I'm not going to get into. But you also, if you have some other sort of the protein has some other function that you can test to make sure that you didn't mess up something else. So if the protein has some enzymatic function, maybe you have a test of that enzymatic activity, you can see if, okay, my mutation didn't mess up that. So I think that it's probably, and I have this nice peak, so my protein's probably fine otherwise. And so this probably likely is a site that's actually involved directly in the binding. When you're doing mutagenesis, so if you start with like a structure and you have an idea of where you think it binds, and you can test that directly, you can test that specifically. If you have no clue, like we talked about, we can do some of those truncations, making sure that they don't affect the protein folding and that sort of thing. Oh, another technique for measuring protein folding is there's various forms of light-based techniques that can tell you about like basically the overall shape of the protein, but that depends on you having machinery that can do that sort of thing. I'm not going to get into that here. But okay, so if you don't know where the interaction is happening, or say you think you do a truncation, you know it's in this region, you try to find the smallest possible piece that will still bind it, and then you can change every residue in that piece. So typically what you do is like an alanine scan or whatever. So you can change all the residues to alanine, and so alanine is kind of like the generic amino acid. So when we talk about amino acids, alanine is kind of this generic nonpolar pretty small, but not like glycine. So it's the second smallest, glycine is the smallest, but glycine is super flexible because it's so small, its side chain is just a hydrogen. And so alanine has this methyl group. And so we often do change things to alanine if we're trying to see if a various amino acid residue is important for a function. So you often see like an alanine scan. There's also like high throughput manners of like deep scanning, where you change to different amino acids, all sorts of things, and the high throughput manner. Not going to go into that, but that sort of thing does exist. But typically, I'm working with things where we have some sort of idea that a specific amino acid is important. So often, we even might hypothesize about what part of the amino acid exactly is important. So maybe we think it is the charge. So charged proteins can form these like salt bridge interactions with one another, where you have like a positive and a negative charge. So maybe we think that something like this is happening. What we can do in that case is, instead of just changing it to alanine, if we want to see is the charge itself important, we want to change to something that's more similar in size. So we can change, for example, a glutamate to a glutamine, or an aspartate to an asparagine. We can also flip the charge, which should totally mess up the reaction. But if we flip up both of them, then we should get it back. And so there are different things that we can test. And in this case, we want to test the smallest thing possible, the smallest difference possible, in order to see whether that specific thing was important. You might also think that, for example, the OH group is important. Maybe you think it's forming a hydrogen bond. In this case, you can change like a tyrosine to a phenylalanine, or you can change a serine to an alanine. So these various things, the basic idea is that you want to make as small as change as possible in order to minimize the like off effects or the indirect effects of that sort of thing. And if you have an idea of what specific thing you think is important, the more like granular you can get, the better. And so then you can use your mutated protein and test for nucleic acid binding, or for like protein-protein binding type of things. And so we haven't really talked about the protein-protein interaction, so let's talk some more about that. So a few main ways we'll do this. We talked before about how there were there were some biophysical techniques. So a lot of them you can use for protein nucleic acid, for nucleic acid, for protein, you can use different types of molecules. Some of these require labeling, some of them don't. So we talked before about SPR. I've tried SPR once. I was trying with like RNA sticking to the chip, and I was having problems with like the protein getting stuck on there. Tried a lot of optimizing, but never got it really to work out, and then just kind of moved on because I had other techniques that would work. You also can like tag your protein and attach the protein. So there's different things that you can do. It might take troubleshooting, and some of the chips you can like reuse, but there's various complications, various details, and a lot of different stuff. But SPR can be a really great technique. I just haven't used it that much. You can also use like ITC, isothermal calorimetry, which is going to measure the like the energy given off when these molecules interact. And it's going to tell you about like thermodynamic properties. Another couple ways we'll talk about are native page and analytical size exclusion chromatography. So we talked about chromatography before and how we could use size exclusion chromatography to separate proteins by size. If you have protein that is bigger, it's going to elute earlier because it's not going to have to go through all the tunnels, so it's going to come out earlier. If you have something that's small, it's going to come out later. If they're interacting, now you have something that's bigger than both of them, and so you're going to get your peak shifted to the left, and this is going to tell you about that they're binding together. So we can use this like analytical size exclusion chromatography. A downside of this is that you need your proteins to be really pure. and you need them to have, you need a lot of protein typically in order to get these nice peaks that you can see and do all your controls and various things. We have some like analytical size columns, which are smaller, don't use quite as much, but still you can be, if you don't have that much protein, it's not going to be a great way to go. And if this is relying on this complex being stable enough that it can go through this column without coming apart. And so if you have a weaker interaction, they might not form a stable enough complex for this to actually work. Another method you could use is a native page. And so basically with the normal SDS page, you're denaturing the proteins and you're coating them with this SDS detergent to keep them unfolded. And so if you're unfolding these proteins, you're going to disrupt their interactions. If you don't unfold them, then you can run them as is. So the complexes, if they're strong enough, are going to stay together. And so in a normal, so when you run this native page, they're going to stay together. And then you can do things like Western blot or mass spec to see if they're actually bound together. You can look for the higher bands, that sort of thing. So because you're now not having this negative charge, so sometimes you need help with getting your proteins to run. There's a technique I've tried called like blue native page, which uses like Kamasi dye to kind of help things along, and I have a post on that as well. So I hope that helps you kind of just get a sense of how you might think through some of these things, maybe give you some ideas for your projects. And yeah, so I hope this helps, and happy learning and studying and discovering new things because you don't know until you test.